Okay, so we'll go ahead and get started with information from Chapter 1. Chapter 1 is just an introduction and an overview of what we're going to be covering. Biology is your study of life, and we're studying just a really small portion of biology, the cells in here. Evolution is the process of change, and it's transformed life on Earth. This book likes to look at different life forms and say, this is what's going on, and try and relate it to evolution and say, this is why it does this and how it got to that point. So it's one way of looking at things and studying biology. I opted for this book because this is the one that is used by most other schools in the state, and so the more close and consistent information I can give you, the better off it will be if you end up going to any other schools in there. So when we look at life, one of the things we know is that it always has ordered structure. It's not random. You don't just get randomly all of your body parts. We all have eyes up here, nose here, mouth here. We don't have some people with an eye here and one on their backside. It's ordered, and it's the same in everyone. Evolutionary adaptations will occur, so if the environment changes, the organism attempts to adapt to it over time to be able to survive in that area. So we have responses to the environment or change. If I were to make this environment in here, turn it up so that it was 90 degrees, most of you would probably start to sweat as a way to try and adapt or change with your environment. There's reproduction. Living things need to create new living things to keep it going in there. We have metabolism, which is basically just chemistry going on in a living organism in there. It's your energy processing. And then we have growth and development. So fortunately, we are not born as full-size organisms in there. No one would really want to have to give birth to a full-size human being in there. We grow and develop with time. And even single-celled organisms will usually start out smaller and grow and develop over time. And biology tends to have a hierarchy that extends from microscopic scale organisms to the entire living planet. Each time you move up in the hierarchy, we tend to see emergent properties that are new or unique properties at that level that weren't present at the level before. So things will, as you move up the hierarchy, become more complex in there. So we are higher up on the hierarchy than, say, a worm, and so you're going to be more complex than a worm is. Your body systems and your body parts are more complex than the worm. When we study things, we tend to use reductionism, where we will take this whole complex system and break it into simpler parts to study. So when you study the human biology in there, we don't study the whole body all at once. We will tend to break it down into systems, and you would study the skeletal system or the muscular system or the cardiovascular system. And that can be useful. It makes it into bite-sized pieces, but there's also limits because it can't fully explain what's going on at higher levels in there. The example they give is Watson and Crick. They discovered the molecular structure of DNA, but that's all they did. They described the structure. They did not describe what it does, how it interacts, anything like that. So if you're just studying the skeletal system, there is going to be some stuff that gets left out. How it interacts with your muscular system, how it interacts with your kidneys, everything else. And all the systems do tend to interact together in there. So we have to balance reductionism with a holistic way of looking at things, where we step back every once in a while and look and say, how does this fit into the whole big picture? So systems biology is the most common way of breaking things down, where we study the components and look at how they function together in the various organisms. So organisms are going to interact with their physical environment or their ecosystem, and their ecosystem is actually going to be affected by the organisms in there. So with plants, water and minerals are going to form in the soil for the plants to eat, but the plants also help to break up the rocks in there with their roots to put more minerals back into the soil. So it's a mutual relationship there. Carbon dioxide gets taken into plants and then they release oxygen. The animals, on the other hand, are going to take in the oxygen and release the carbon dioxide that the plants need. When you have insects, they're going to eat the plants. Other things eat the insects, and then we eat those other things in there. Sometimes insects are eaten, plants are eaten by humans. And eventually we die, and then we're broken down and decompose and put minerals back into the soil. So for some reason, humans are kind of obsessed with avoiding putting themselves back into the cycle. We like to put ourselves in a box and keep all of our minerals for ourselves, where pretty much every other organism that dies fully decomposes and goes back into the soil at some point. Humans seem to just want to take from the planet and not return themselves back in. I don't know why. But the nutrients are going to cycle throughout the system. Everything goes back to where it came from eventually. So when we see global climate change, it's going to show an increase in temperature and weather patterns around the planet. One of the things with the global climate change is they always talk about global warming, and when you live someplace like here, you don't really get warming. So I've lived in Washington way too long, and I can tell you our climate really isn't getting any warmer here. However, what we are seeing are more extreme weather patterns. So in some places you see warming, but in our particular range of latitude, we get more extreme weather patterns. So global warming is not going to turn Washington into the tropics, no matter how long you wait. What you'll get is more storms. So when it rains, it's going to rain harder, more of it, and things will just become more erratic. So with living things, we have energy that gets transferred and transformed. At the lowest level, we have the producers. These are going to be your organisms that are going to absorb light energy, and they end up transforming it into chemical energy. So these are organisms that can do photosynthesis. It's going to be your plants. So they'll take light and transform it into sugars to make their structures. Also some of your algae and bacteria that do photosynthesis. And it's thought that they had to be here first to create the oxygen in the atmosphere for the plants to be able to eat later on. So the energy of those chemicals that are made from photosynthesis ends up being transferred to the consumers when they eat them, who use it to do work in there. So consumers feed on the producers and other consumers. We are consumers in there. If you don't do photosynthesis, you're a consumer. When we look at the different parts of the cell, what we find is that each of the parts has a function in there. And when you look at it, a lot of times it makes sense to have that part, and their functions are actually logical in there. If you look at it from the point of view of a cell, it has a reason for what it does. And to me, that makes it easier to remember. If I can look and understand, well, why would it want to do that? Then I can remember it. If I look at it and go, that seems like a completely random thing, it's much harder to remember. Some of the cells are going to have some common functions that will be present in all of the cells in there, and so they'll have some common structures that will allow that. All cells are going to be enclosed by a membrane. The membrane is going to control the passage of what goes in and out of the cell. It's going to hold the genetic material in the cell. It's just going to be a barrier between the cell and the outside world. And our cells fall into two major categories. You've got the eukaryotic cells and the prokaryotic cells. So your eukaryotic cell is going to be the bigger one here. It's got a membrane around the outside. This one has a membrane around the outside. Eukaryotic cells have genetic information. Prokaryotic cells have genetic information. However, the eukaryotic cell is going to have these internal membrane spaces that are going to separate the nucleus and the organelles, where the prokaryotic cell does not separate things. So you can kind of think of it as, this is a purse that has all kinds of different compartments for your stuff. This is when you throw it all in the shopping bag. and it's all randomly in there. They still have the essential parts, it's just one's more organized than the other. So all cells are going to have some continuity with heritable information. So it's going to take some traits that it has and it passes it on to its offspring. And it's going to do this through your DNA or deoxyribonucleic acid. Your genes are made of DNA, and the genes are the units of inheritance that are going to code for the proteins that make up your structures. So when we look at the DNA in there, it's going to form chromosomes when cells divide. The chromosome is what you most likely think of when you think of DNA. So those are those X-shaped things like this. So your DNA is this double-coiled helix, but that keeps folding itself up until it can make this X shape in there. The genes are just your instructions to build a protein. So a gene is going to be a particular region of the DNA on this chromosome in there. So the gene is a much smaller part of it. The proteins are going to be what actually create the structures and help to carry out cellular work, give the cell identity. So the majority of your structures are made up of proteins in there. Proteins are not really what your cell wants to use for energy. It is definitely used for structure. So the chromosome is going to carry then all of those instructions, and when the cell replicates, you're going to copy the chromosome in there and give it to each of the new cells so that it got instructions from its parents on what to do. And it acts as your central database for everything the organism does. So all of your cells have all of the instructions to make everything in your entire body. You just don't use all of the instructions in all of the cells. So there's no need to make saliva with your hands. You just turn those instructions off. So your DNA molecule is a double-stranded helix, and it's made up of 4 nucleotides in there, A, T, G, and C. So it's kind of shaped like a ladder that is twisted. So if it's twisted, it's just twisting it around. And so you're going to have A, T, G, and C nucleotides that are going to make up half of the rung of the ladder. And then the other half will be made up with complementary nucleotides in there. And we'll go more into that later on. So when we say we're expressing a gene, that's when you're going to actually make a protein or a product from that gene in there. We call it being expressed when you're making something from it. And you don't use the DNA directly to do it. You're going to use an RNA intermediary in there to make that. So it's kind of like if this is your master set of instructions in there, you're building a house, this is your master set of plans. The person who's pouring your foundation isn't going to get that master copy of the master set of plans. They're going to get a photocopy of it in there so that all the different workers can get the photocopies in there. And the photocopy is the RNA intermediary that we use. So in the last 50 years, studying the genome has become a whole separate field in there. The genome is your entire library of genetic instructions that the organism inherits. It's a very generic term. It doesn't imply that it's in any particular stage or shape. Chromosomes, these are just when the cell is dividing. But most of the time, that's loosely coiled. I like to think of this as like when you're moving and all your books are packed in boxes. They're all put away, all the information's there. It's just not very accessible to you. When you're actually using your books to study, everything's all spread out, the pages are open and you can get to them. That's the genome, where it's all spread out like that. So we have sequenced the entire human genome in there. That was something they thought they would still be working on for at least another 10 years from now, and they were able to computerize it and get it done early on. I believe it was in 2002 when they finished it up in there. And you can actually go online and look at the sequence of the human genome if you want to. It's just a bunch of A's, T's, G's, and C's, so it's not real exciting to look at in there. But by sequencing so many genomes now, we have a lot of computer information of those. And so they've used computers to be able to analyze the information with comparisons of what we have in there. And that's a whole new field, the bioinformatics in there. With high throughput technology, we're able to use computerized tools to analyze the biological materials really rapidly. So it used to be if you wanted to analyze the DNA of something, you had to collect it. It was going to be a couple days to process it in there. You were going to have a couple days of amplifying the DNA, making copies of it. You had to do that all by hand. You had to run gels on it by hand and then analyze them by hand. And for the most part, now they can collect it. If there is just a small amount of the DNA and they need more of it, they can put it into a machine that does the PCR. It comes out amplified in a few hours automatically. So it used to be that you stood over it with a timer in this water bath and you would put it in at one temperature and wait. When the timer went off, you would add something else in it and stick it in at another temperature and wait. And then the timer would go off and put it in a third thing and do it. And you had to just keep manually repeating that cycle. And now you can just stick it in a machine, push a button, and it goes like a microwave. So another thing we see in most living systems is these feedback mechanisms in there. Living organisms like to maintain homeostasis, or keeping a constant, stable environment. If you've ever tried to lose weight, your body will show you how good at this it can be. One of the things that if you eat less, it's going to say, oh no, I don't want to part with any of my precious tissue in here. I'm going to slow down your metabolism. I'm going to make you hungry so that you want to eat something even more. It's going to pull out all its little tricks to try and keep you exactly the way that you are in there. And that even happens down at a microscopic level in there in a cell. One of the ways it will do it is the negative feedback loop. If it detects a change, it tries to reverse it in there. This is the most common process in here. This diagram is showing that if you have this pathway going on where you make A to B, B to C, C to D, if you start to build up too much D, what will happen is it will actually block the pathway and not let it go through anymore. It says, you know what, there's too much of you, stop making it in there. Positive feedback loops, these are going to detect a change and they will tend to reinforce it. These are far less common in the human body. One example is blood clotting. So normally you want your blood to be thick enough that it doesn't leak through and cause you to have bruises because it leaks out your blood vessels, but you don't want it so thick that it forms clots and gets stuck. However, if you spring a leak in the system, you do need it to thicken up in there, and it will do a cascade of events in there to cause it to get thicker. Also, uterine contractions during childbirth tend to work on a positive feedback loop in there. They're going to build in intensity. If they didn't, the baby'd never come out. So any questions on the feedback loops? So with evolution, this is going to be the way that we explain the unity and diversity, so how things are the same and different in organisms. And what it says is the organisms that are present today are descended and modified from common ancestors. So you may have had one ancestor that its offspring diverged into two different pathways to make two different organisms in there. With life forms in there, so far we've identified about 1.8 million different species in there. Thousands of new species are identified every year. It's estimated that we have between 10 million to over 100 million different species. And you may look and go, what, are they really finding that many new insects? You've got to remember when they include all of these things, they're not necessarily organisms that you can see. You know, are we going to find any more cool big mammals? Probably not. We've been through most of the planet on there. However, there are still lots of microscopic things that we can't see with our naked eye, so we don't even know we're not seeing them in there. And those kind of accidentally get discovered out there. So it's an estimate to say there's between 10 and 100 million different species. We don't really know. With taxonomy, this is a way of naming and classifying the different species out there. And they do this based on your shared characteristics. And so as you start out with your domain, which is going to be a very broad, and you can see what by the big piece of the pie on there. It's a very broad generalization descriptions in there. As you become more specific with the descriptions, then it moves down to kingdoms, then phylums, class, order, family, genus, and species. So that by the time you get to species, those are all going to be very similar in there. So it starts out there, it's classified into, there's 3 domains of life. And this is based on the DNA sequences. You've got your bacteria, you have your archaea and the eukarya. And so sometimes this part is a little confusing to people. So we have your prokaryotes and eukaryotes. The prokaryotes, you've got your bacteria and your archaea, and then you have your eukaryotes in there. So these are the 3 domains. 2 types of cells make the 3 domains of life, and they determine that based on looking at DNA sequences in there. So your archaea, these are kind of a weird little group that you don't get to run into all that much. They are almost as similar to eukaryotes as they are the bacteria, even though they're prokaryotes in there. They tend to live in really extreme environments. So these are the things that you would find like in volcanic vents or deep in the ice. They'll live in weird chemicals in there, you know, like in something like sulfur or other things. They're not things that are going to live on us. We don't really know of any of them that cause diseases for us because we just don't interact with them that much. Your eukarya, these get divided into 3 more domains in there that are going to be multicellular. So these are multicellular. You're going to be your plants, animals, your fungi, and then with your, or I guess it's only 4 of them, maybe 5 on there. And then you've got your protists. These are single-celled in there, in the subdivisions. So animals, they get their food by ingestion. Fungi consume dead matter. Plants do photosynthesis. And then the protists, they do a variety of things in there, but they're single-celled organisms. So I think that's a good spot for us to stop today.